So today we have the distinct pleasure of Doctors Olga Gornik and Dr. Najda Rudman joining us from the University of Zagreb, Faculty of Pharmacy and Biochemistry. And the title of the talk is N-glycosylation and its genetic regulation in type 1 diabetes. Just a quick little intro. Professor Olga Gornik is the head of the Department of Biochemistry and Molecular Biology at the University of Zagreb, Faculty of Pharmacy and Biochemistry. And she's leading a team of scientists who explore the role and biomarker potential of N-glycosylation in diabetes. Recently, thanks to Professor Grant Morahan, who is a leading expert in type 1 diabetes from the University of Western Australia in Perth, their collaboration with the University of Copenhagen enabled them to analyze N-glycosylation of plasma proteins and IgG in 2,000 children with recent onset type 1 diabetes from Dan Diab kids. Dr. Najda Rudman is a postdoctoral researcher at the University of Zagreb. She obtained her PhD using these results. The study exposed the biomarker potential of N-glycom in type 1 diabetes and identified novel points in its genetic regulations. They are now eagerly continuing their research in type 1 diabetes, aiming to A, test predictive value of glycans and risk assessment of unaffected individuals using samples from India study, and B, identify the B cells N-glycom characteristic for the disease. As we know, it's been characterized in T cells. And finally, C, they would like to, or they aim to study differential potential of N-glycosylation in different autoimmunity to hopefully contribute to understanding diagnosis, management, and possibly prevention of type 1 diabetes. Their recent excellent paper, Children at Onset of Type 1 Diabetes, showed altered N-glycosylation of plasma proteins and IgG, was published in Diabetologia 2022 August. And so welcome both Drs. Gornik and Rudman. Thank you very much for joining us.
Thank you for your invitation. Yeah.
Thank you for invitation.
Great. So yeah, so take it away.
Okay, thank you for your kind introduction. So as you all heard, we come from the University of Zagreb. Zagreb is capital of Croatia, and the main research field of our group is the field of glycobiology. So actually we explore biomarker potential of N-glycans and their involvement in development of different diseases. So for those who are maybe not familiar with this term, N-glycosylation is a complex enzymatically regulated process by which complex oligosaccharide structures are added to lipids and proteins mainly in our body. It is also important to mention that contrary to proteins, there is no direct genetic template for glycans. So the structure of glycans is actually written in the complex network of genes. And of course, environmental factors also influence their final structures. As you can see, just a second, I need to close the windows. Yeah, as you can see it on the right part, these are just the examples of different glycan structures. These structures can differ in the composition of monosaccharide units, degree of branching, complexity, type of linkages that are present between different monosaccharide units. It is also very important to know that glycans are structural components of nearly all proteins in our body, and not just proteins, lipids are also glycosylated. And recently it was also discovered that non-coding RNA also carry glycans. Here in this illustration, you can see that the surface of our cells are covered with thick layer of glycans, and this dense thick layer is called glycocalyx. So the glycans are actually the first molecules that comes in interaction with other cells, pathogens, other molecules, etc. But not only membrane proteins are glycosylated, almost all circulatory proteins are glycosylated. So this is the example of alpha-1 acid glycoprotein. And from this picture, you can see that it possesses 5 different glycosylation sites. Each of these sites can be occupied with different glycan. And what is also very important, that at the same site, different structures can be present, resulting in different glycoforms of the same protein. And these differences actually carry the great biomarker potential of glycans, since these glycan structures are very sensitive to all pathophysiological processes that are occurring in cells. So today we know that N-glycan is changed in many diseases, and it is also important to mention that it is very stable within an individual during the period of homeostasis, so it adds to its diagnostic potential, and the changes in glycosylation occur very rapidly when something is wrong in the cell. So this all gives great potential to glycans in prediction, stratification, and differentiation in many different diseases. Above that, glycans possess great inter-individual variation within population, and that makes them great potential tools in modern personalized medicine approaches in medicine. And this is why in 2012, even a US National Academies recognized glycoscience as a field that will be needed to realize the goals of personalized medicine, and that glycans are directly involved in the pathophysiology of every major disease. Maybe for this audience and this topic, it is important to explain that glycosylation complexity is an evolutionary process. So glycans present on the surfaces of pathogens are different than that in higher organisms such as mammals, and this difference actually is the basis of molecular sensing and self-non-self discrimination. Both innate and adaptive immune cells are equipped with a variety of glycan-recognizing receptors such as galactins, C-type lectins, Siglex, and etc. And when they recognize the different glycosylation forms on the pathogens, they initiate either pro or anti-inflammatory responses. Glycans can also modulate the activity and function of immune cells, which I will talk a bit later in context of type 1 diabetes, as well as the effector functions of antibodies. When it comes to antibodies, of course, immunoglobulin G is the best studied glycoprotein. For long, it is known that it has one glycosylation site at FC region, that's on asparagine 297, where biantennary glycan is attached, and over maybe, I don't know, for over the last few years, scientists are also aware that about 20 to 30% of molecules of our IgG also are glycosylated at FAB part. And although we actually carry very similar structure, or better to say the same type of glycan, biantennary glycan, it is important to mention that these glycans differ in monosaccharide composition, and with this glycan on FAB region being more sialylated. So there is much more glycans bearing sialic acid and, for instance, bisecting LCNAC here on FAB region than glycans on FC region. IgG, it is also important that IgG effector functions are controlled by these glycans on FC region, and they define whether IgG will have a pro or anti-inflammatory function. For instance, the absence of this core fucose at the core of the glycan actually increases ADCC, while addition of sialic acid at the end of the glycan reduces affinity for FC gamma receptor and makes antibody anti-inflammatory. So this is important maybe to have in mind while performing glycoengineering, for instance, of intravenous immunoglobulins, etc. I also wanted to mention here that in some of the previous talks at Sugar Science, I noticed that, for instance, I think it was Dr. Jacobson, that she was searching for, let's say, potential biomarkers to follow the response to immunotherapy. So maybe glycosylation of IgG could be one possible biomarker or one possible target. When it comes to glycosylation of FIB region, now we also know that glycans present on FAB region influence the function of IgG. As expected, their most important role is in antigen binding, where steric or charge repulsions occur due to negatively charged salicylic acid at the end of these glycans, and that influence the affinity for antigen binding. N-glycosylation of IgG is changed in many different autoimmune diseases. Here is the example of the best studied rheumatoid arthritis from the side of glycosylation. And here is just the example from which you can see that glycosylation of IgG changes during different stages of the disease, and it can be used to monitor disease activity. So over the last few years, by our group and others, it was shown that N-glycosylation is involved in diabetes development, and not just type 1 diabetes, but also type 2 and monogenic diabetes. But glycans are not only involved in different pathophysiological processes behind the development of these diseases, but they also possess great biomarker potential in differentiating between different diabetes subtypes. But today, type 1 diabetes is the most interested to us. So I just wanted to share some previous knowledge on the importance of glycosylation in this type of diabetes. As I already mentioned, glycans influence T cell activation. And how do they do that? There is one enzyme called N-acetylglycosamine transferase 5, which is actually responsible for the formation of this complex, highly branched glycans. And when these glycans are present on these T cell receptors, then they limit the TCR mobility and clustering. The galectin-3 binds to them. For lattice, formation of lattice occur, and it also increases the retention time on cell surface. Contrary to that, loss of MGAT5 expression, so the loss of this complexity in glycosylation, when we have TCRs glycosylated with much less complex, so let's say these smaller glycans, it results in their clustering and T cell activation. So actually it decreases threshold of T cell activation and hyperimmune response, and increases susceptibility to autoimmune development, to autoimmunity. Also, many genes for different glycosyltransferases were identified as connected to type 1 diabetes. Maybe the best studied example is the FUT2 gene. It's a gene for glycosyltransferase that adds alpha-1 to fucose to the galactose on glycans, which was identified as one of the causal candidate genes for type 1 diabetes. And it is also known that N-glycosylation is associated with complications in type 1 diabetes. Here is the example of the association of glycans with renal complications in patients with type 1, adult patients with type 1 diabetes. But actually the cross-sectional study in type 1 diabetes from side of glycosylation has never been performed before. So thanks to Professor Grant Morahan and Fleming Poisson, we actually got into possession of plasma samples from almost 2,000 children from Danish Register of Children and Adolescents with Diabetes, and 200 of their healthy siblings. We analyzed N-glycans of their total plasma proteins, or IgG, and we identified differences, as well as afterwards identified gene variants that are responsible for these differences. But since this was the part of the PhD thesis of my previous doctoral student, now postdoc Najda Rudman, I would like her to present. She would be the best person to present this study and its results.
Thank you very much for that introduction. Yeah, go ahead, Najda, and take over.
Okay, do you see my slides?
Perfect.
Okay, so first I would just like to warmly welcome everyone listening to this talk and send our greetings from faraway Croatia. Today I'm delighted to present and discuss with your audience results generated throughout my PhD studies. And just before starting with these results, I wanted to share a quick overview how N-glycosylation is usually performed. So glycans are complex, non-linear structures, and their analysis was challenging. We still can't look specifically at the glycoprotein, but instead we mainly focus on glycopeptides or on released glycans. And then we study those glycans usually by liquid chromatography or by capillary electrophoresis. Or for example, when we cut glycoproteins into glycopeptides, we usually then analyze them by mass spectrometry. So to date, we developed high throughput methods for plasma protein N-glycosylation, for IgG N-glycosylation, and recently we also developed these methods for alpha-1 acid glycoprotein and for complement C3 protein. So on this slide, you can see how the output of these analyses looks like. So actually here is represented this representative chromatogram of plasma N-glycon, and on the bottom part of the picture, N-glycon of IgG. So if you take a closer look, you can see some differences in their glycan composition. So for example, for plasma glycans, we can identify very complex, highly branched structures, whereas within IgG N-glycon, those structures are not present. So the most complex IgG N-glycan is actually decellular IgG N-glycan, biantennary decellular glycan. So also after these analyses, when we get our data, we need to perform also some complex steps in order to statistically analyze this data. So we usually do quality control. We do, for example, correction to remove technical variation. We transform our data to normal distribution, and usually we correct this data for age and gender, because in many studies of the general population, it was determined that glycosylation is associated with gender and age. Now I will finally start with the part that concerns N-glycosylation changes of plasma proteins and IgG and its genetic regulation in recent onset type 1 diabetes. So our collaboration, as Olga already mentioned, with the University of Western Australia and University of Denmark, resulted in novel insights into the glycom and genome interplay at the onset of this disease. And these results were recently published in one of the leading European diabetes journals, Diabetologia. So this was actually the first study of plasma protein and IgG N-glycosylation at the onset of this disease. And this study, as Olga already mentioned, included almost 2,000 children with new onset type 1 diabetes. So their plasma samples were taken within 3 months of the disease diagnosis. And also for a subset of this population, we received samples from their healthy siblings. So if you can see from this slide, our study was actually divided into the primary study, which comprised only diseased children, so almost 2,000 children with the disease, and follow-up family-based study where we had SIB pairs, affected and unaffected SIB pairs. And when we obtained these N-glycosylation data of this cohort, we use this to check for differences between children with recent onset type 1 diabetes and their healthy siblings, and later to construct type 1 diabetes discriminative model. So our results show that the onset of this disease is actually associated with increase in some very simple N-glycans. So here we observed increase in high mannose glycans, some glycans which terminate with N-acetylglucosamine. We also observed increase in IgG decellulation, and we observed decrease in monogalactosylation. So based on these results, we constructed type 1 diabetes discriminative model, which included age, sex, together with N-glycans. And these models yielded notable discriminative power between children with recent onset type 1 diabetes and their healthy siblings, with area under the curve of more than 0.9. for addition of plasma N-glycans and almost 0.9 for addition of IgG N-glycans. So what is also very important to mention is that in our previous study of adult patients with type 1 diabetes, we identified completely different changes in their N-glycon. So, for example, when we studied these adult patients with diabetes complications, actually we identified a shift toward very complex glycan structures. And if you remember, in these recent onset type 1 diabetes children, a shift in different direction was observed, so toward more simple glycans. So this was really important to us because it may show that glycans may be involved in different steps of this disease. And also the glycans are not just a reflection of glycemic changes. As Olga already mentioned, on some glycan parts of FAB and FC portion of IgG and how it influences immunoglobulin effector functions and antigen binding. I will just briefly again mention those glycans that were revealed to be changed in children at the onset of this disease and how they might actually influence the IgG functions. So if you remember from Olga's talk that on the FAB fragment of the IgG, so the fragment that is responsible for antigen binding, most commonly the glycan is decellular. So in our study, we observed increase in this specific glycan. So in children at the onset of this disease, IgG decellulation was increased. And Olga already mentioned, I will just briefly again go through it, that this sialic acid on this glycan is negatively charged. So due to charge repulsion between these specific glycans and antigen, it at the end affects affinity for antigens of this fragment. Also, what was also a very interesting observation at the onset of the disease was increase in bisecting GluCnac structures. And from many previous studies, it was shown that bisecting GluCnac is associated with increase in IgG ability to destroy target cells through antibody-dependent cellular cytotoxicity and through its affinity to Fc gamma receptors. So this was important to us because it is considered that one of the main triggers of type 1 diabetes, one of the triggers could be possibly virus attack or pathogen attack. So this could be the process that is involved in fighting with these infections. Also, for IgG glycans, it was demonstrated previously that that changes in their specific change in one specific structure of this glycan is associated with increase in complement activation. So, for example, if you have terminal GluCnacs on these IgG structures, and if you remember, I also mentioned that we identified increase in these structures that terminate with GluCnacs and also in structures that terminate with high mannoses. So in previous studies, especially involving rheumatoid arthritis, it was shown that actually IgG with those kind of structures activate complement activation through mannose binding lectin pathway. So before this study, it was not known that IgG can activate this part of the complement pathway. It was known that IgG can activate a classical pathway of the complement activation, and this is also through glycan interactions. So glycan interacts with the C1q of the complement and activates a classical pathway. But in rheumatoid arthritis, it was shown that IgGs with terminal GluCnacs activate mannose binding lectin and complement activation and contribute to joint inflammation in rheumatoid arthritis, and even can precede the disease onset, and they can later stratify the patients. So many of these studies were done on rheumatoid arthritis. This large cohort also enabled us to study N-glycosylation differences which are associated with gender and age. So actually, we can also say age at diagnosis, because these children that we studied were at the beginning of their disease, so when they were diagnosed. So within 3 months of the diagnosis of their disease, we have their plasma samples of that time point. So significant N-glycosylation differences between genders were mainly observed upon onset of puberty, which was expected since we already did many different cohorts, glycan cohorts in general population, and we already observed hormonal influences on glycosylation. However, some of the derived traits, which were significantly different between these studied groups, changed even before the onset of the puberty. This was also very interesting to us. For example, those bisecting look-like structures on both IgG and on both plasma proteins changed differed between genders even before the onset of puberty. And also, we think that it is important to examine these differences, since type 1 diabetes may also have a hormonal component, since it was demonstrated that after the onset of puberty, male gender is more at risk of developing this disease. But regarding these results, it is somewhat hard to comment on it, because some of the disease-associated, risk-increasing associated glycans were decreasing in one gender, and some risk-associated increasing glycans were in different directions. So here it's a little more complicated to comment on this part. After this N-glycosylation analysis, we performed genetic association studies. So this was also the first genetic association study on glycosylation data in type 1 diabetes. And for a total of 1,105 children with type 1 diabetes from this registry, then DiabeKit registry, we obtained almost 200,000 SNPs. So we identified almost 200,000 genetic markers in each of these childhood cases. And we genotyped these children with immunochip. And then we tested these genetic markers, SNPs, with variable levels of IgG and plasma glycans. Okay, so what we got as a result was that a novel association was uncovered for N-glycosyltransferase MGAT3 and specific IgG glycans. So in the general European population, so the largest actually genome-wide association study that was done in the general European population comprised around 8,000 individuals. And from these data, never before these specific SNPs that are within the candidate, the MGAT3 gene, were associated with this specific glycan. And if you remember from the first part of the talk, this specific glycan was that DC-allylated glycan, which was increased in type 1 diabetes recent onset cases. So here we identified genetic variants contributing to these changes. Also, what is interesting is that in the general European population, this specific glycan was never before associated with this gene, but was associated with one different N-glycosyltransferase, which is ST6GAL1. And this transferase encodes protein that is responsible for addition of sialic acid. And also another interesting thing was that in this study on recent onset type 1 diabetes cases, most significant genetic associations were with MGAT3 gene. And in the general European population, most significant genetic associations were again with ST6GAL1. So it seems that something is happening here and is changing the regulation of these two N-glycosyltransferases. And I will later comment on this and why do we think this is happening. Okay, so regarding the plasma N-glycosylation, we identified novel genetic locus associated with plasma N-glycosylation, so complement component C3 gene. So in the largest genome-wide association study on N-glycosylation data, this gene was never before associated with plasma N-glycosylation. So what is also very interesting is that missense variant in this gene was associated with levels of high glycan. So usually in genetic association studies, usually those variants that are identified are in intergenic regions. So here we identified variants that causes amino acid substitution within this gene. It is a missense variant. And this variant was associated with decrease in one specific plasma glycan, so high glycan. So if there is a change from cyclic to acyclic amino acid within the C3 gene, within the C3 protein at the end, there is a decrease in levels of high glycan. So we searched through the literature trying to explain this association, and we found out that the C3 gene product, which is a complement C3 protein, the main protein of the complement activation pathway, has such associated glycan on its surface. And this glycan is actually located at the domain which is included in pathogen binding of the C3 protein. So this was also very interesting to us. And also we found out that from the literature that this specific variant that we identified is in high linkage disequilibrium, which means that it is often co-inherited with another variant which was previously associated with type 1 diabetes risk. So this variant from the literature was identified in TEDdy study. And also our identified C3 variant was previously associated with altered complement activation. So it was actually associated with increased alternative complement activation pathway. And we really believe that this may be important for the onset of this disease. Its complement was associated with some of these processes happening at the onset of the disease. Also, we searched through the literatures to check for possible pleiotropy of these identified variants with other gene or protein expression. So these variants that we identified, I'm sorry, just a moment. So the variants that we identified in the MGAT3 gene, so in the candidate, so variants that we identified were not for MGAT3 gene, were not within the gene, but in the intergenic region close to N-glycosyltransferase MGAT3. So if you remember, I already told that usually genetic association studies usually identify variants in intergenic regions. And then later you have to search for candidate genes responsible for these changes. And here it was biologically plausible that MGAT3 is the candidate gene because this variant was close to MGAT3, and MGAT3 is responsible for the formation of such associated glycans. And also what was interesting is that these variants in MGAT3 gene are associated with, within the candidate MGAT3 gene are associated with increased MGAT3 expression in B cells. And B cells, activated B cells are IgG producers. So this was relevant because this explains why we might see these associations. And also one interesting thing was that these variants were associated with increased interleukin-6 receptor B expression in plasma. So this interleukin-6 receptor B is also known as soluble glycoprotein 130, and it inhibits interleukin-6 trans signaling, and enhanced T-cell responses to interleukin-6 were previously associated with early clinical disease. And also one other thing which I already mentioned is that these variants that were identified within the C3 gene, so in this case, really within the gene, not in intergenic regions, so within the C3 gene are in high linkage disequilibrium with another variant previously associated with risk of developing type 1 diabetes in the TEDdy study among those individuals that are having one of the highest risk genotypes associated with this disease. With having these results in mind, we are continuing our research. We hope to identify molecular mechanisms of these changes and possible impact of these changes on type 1 diabetes risk or progression. And Olga will now just finish our talk.
Okay, sorry, thank you. No, I don't have anything to add about the topic. I just want to thank all. So I wanted just to thank all our collaborating institutions and funders, and also the people who recognize the importance of this work and are now willing to join our research. As it was already previously mentioned, we are now analyzing glycosylation in India samples, so trying to see what is going on prior to disease onset, so following glycans from birth to the development of autoimmunity and some similar research. So thank you very much.
Thank you very much for this excellent talk. I'd like to open it up to our audience. If you'd like to ask a question, feel free to unmute yourself. Wants to share a resource. Someone has shared a resource with you guys for the T2DHugeAmp.org.
Yeah, thank you.
Which is had a recent paper sort of describing its impact. Sorry, I don't know. Recording. It's been great, and that's been driven by Noel Burt at the MIT The Broad. So,
may I ask you a question?
Yes, please.
Thank you very much for this talk. And my question is, if you have analyzed some glycosylated hemoglobin in diabetes.
Okay, so yeah, from the side of, you mean just glycosylation or glycation of the, because usually the glycation of hemoglobin is something that is studied in the form
modifications.
Yeah, no, we actually, we do not study glycation. We only study these enzymatically added glycans, enzymatically added sugars. So this is, let's say, our main research interest. But in our studies, in those involving both type 2 and type 1 diabetes, we often correlate our glycosylation results with HbA1c levels. So it was shown actually by others and also by us that the modification in glycans, especially this shift toward the increased branching, is often, let's say, not all, but is often actually in the direct relation. So it's with HbA1c levels. And sometimes it is speculated that actually the increase in the levels of glucose in our blood that starts this sorry, I'm tired.
Yeah, I can also comment.
Hexosamine. Hexosamine pathway. Sorry. Yeah, it's almost 8 p.m. here, so we are working all day. So hexosamine pathway and increased influx of glucose through hexosamine pathway, and the result of this pathway is the UDP GLCNAC, which is actually the substrate for glycosylation. So we are just studying this enzymatic changes, enzymatic additions, but yeah, we correlate them with glycation of hemoglobin, and so that there is relation. And this is also what Najda mentioned, that she saw this connection, that we saw connection with increased glycans adult population with already developed complications, contrary to children who had this let's say lowered branch glycans predominantly present. And these highly branched glycans are probably actually just the consequence of this bad glucose levels, or how to say it,
metabolic
they're
regulation.
not, metabolic, those people, of course, are not always perfectly regulated. So this result in increase of these complex glycans that are not present at the end of the disease. So,
but at the onset of the disease, we observed a completely different change. So towards very simple glycans, whereas in adult patients with diabetes complications, very complex glycans were associated with these glycated hemoglobins. So this is not, something else is happening here. So this is not a consequence of increased glucose or those kind of pathways that can also occur, of course.
And it is also maybe interesting to mention that meanwhile, after these papers were published, we actually developed the high throughput method for the analysis of C3 glycoprotein complement component. So as this was the hit, but we didn't have the tools to study it. So we developed this high throughput method, and we saw that also on this isolated proteins, so on C3, the forms that are mostly increased on the protein are those that were, would say more simple, but earlier in this glycosylation pathway. So yeah, it is also interesting and maybe in some relation to this.
Yeah, that's very interesting. Is there anyone else who'd like to contribute or ask another question? Please feel free to unmute yourself or drop something in the chat. Okay, well, in the interest of time, thank you very much for presenting this wonderful study, and also the fact that this was part of a PhD dissertation is phenomenal. Right, so I mean, that's really congratulations to you both. So
thank you.
I hope that many people will watch this and reach out to you perhaps for more collaborations. And when you're talking about this idea of a potential biomarker, that's very hot topic right now, particularly, as we're understanding endotypes and within type 1 diabetes, and understanding, sort of the pathway, the prodrome pathway to type 1, and how we can either extend that or avoid diagnosis altogether. How can these biomarkers inform that space? So it's a very important topic. And thank you again for joining us. I hope you have a great rest of your evening.
Thank you for inviting us.
Yeah, you too. Have a great rest of your day.
Thank you. You're in Los Angeles. Okay, thanks again. Have a great day. Bye-bye.
Bye.